Live on YouTube now. Yes, the stream is live.
Okay, all right.
And if
Actually, last time, Mansoor, dear Mansoor, actually last time I click on live, but there was a option that was paid, like they was demanding dollars. So
Mm, maybe you click on something in description like when we start live, there is an option of agreement like
okay,
they give some description like you can remove this description, but uh, I will do this later, but no, it's okay. We are
okay.
live
Thank you
now.
so. Yeah. Let me share. मंसूर kindly YouTube पे जाके लिंक शेयर कर दें या लिंक वो उनको दे दें आप
group link,
मिस या मिस
Okay, okay, I will.
ओके मिस अतिया को वो लिंक दे दें वो ग्रुप में शेयर
she
कर
has
दें
shared. She has shared.
ओके ऑलराइट दैट्स ग्रेट सो अस्सलाम वालेकुम एवरीवन आई थिंक we wait just 2 or 3 minutes.
Sure, sure. अच्छा जो मैंने थोड़ा सा पहले स्पीच की है वो फिर
Yes,
सुनेंगे?
yeah. Sir, kindly let me start. Assalamu alaikum, everyone. I hope you all are going well. And welcome in today's first internship class of biotechnology and bioinformatics uh, by the Dr. Shahzad Zafar, who is currently a doctoral researcher at Shenzhen University, China, and his study field of study is biomedical engineering. Now I would like to invite Dr. Shahzad Zafar. Yeah, sir, please go ahead. Thank you so much.
Yeah, thank you so much, sir. Thank you too. Uh, Thank you so much for having me here. Assalamu alaikum to everyone. I am Manish Rajapur. Currently, I'm serving as the Senior Vice President of VitaNova. I would like to thank you, Mr. Ramurtuza, for starting this initiative to.
Sir, sorry to interrupt. Please, I think your voice very low. Please, can you check?
Now it's okay. Now it's okay.
Yeah, no, okay. Thank you so
Okay.
much.
Yeah, sure. And thank you so much to all of you for joining this summer internship program. And I also would like to thank the White Nova leadership who has worked hard to start this initiative. And today my topic is to deliver a lecture on introduction to biotechnology. In this lecture, I will discuss how biotechnology started, how its emerging phases and its branches, and PCR techniques. Uh, let's start today. Let's start. What is biotechnology? This word came from the Greek origin. This word is a Greek origin, and bios means life, and technicos means skill or art. Together, joining this word, the word biotechnology emerged. Uh, if please, sir, someone changing the slides. Yeah, please. I will change the slide. Okay. Okay. Biotechnology is broadly defined as the science of using living organisms or the products of living organisms for human benefits, uh, or to benefit human. Uh, that is to make a product or solve a problem. The word biotechnology was first used in 1917 to describe processes using living organisms to make a product or run a product, such as industrial fermentation. In biotechnology, we will use living organisms or any product, or together with these products used for the benefit of humans. using scientific methods with organisms to produce new product or new forms of organisms. That is totally biotechnology. If we talk about the phases, phase by phase development of biotechnology, is phase one started from ancient or traditional biotechnology before 1800. Before 1880s, the major inventions and discoveries were like fermentation. This was 6,000 before birth of Christ. Beer and wine production in Mesopotamia and Egypt, and bread making 4000 before Christ, the birth of Christ, use of yeast and cheese and yogurt, and some selective breeding were done at that era. Animals and crops like corn domestication in Mexico. Uh, Traditional medicine, if I talk about this medical implication, use of plants extracts to, for example, Ayurveda and Chinese medicine to heal wounds and to reduce the body temperatures. Uh, the phase two is regarded as the classical or cell-based biotechnology. Its era is 1800 to mid 20th century. In this era, there were a lot of inventions and discoveries, but I listed the major, the most prominent discoveries that almost every science student knows. And in this era, Louis Pasteur, 1857 to 1864, discovered microbes in fermentation and pasteurization. Uh, he, uh, he demonstrated that the microorganisms, yeast, and bacteria are responsible for fermentation, not spontaneous chemical reaction. He showed that different microbes produce different fermentation products like lactic acid and alcohol. And in 1862, pasteurization, uh, he developed the process of pasteurization, heating liquids like wine or milk to specific to a specific temperature to kill harmful microbes without affecting quality. Uh, and in 1864, the germ theory of diseases emerged, and in under this theory, he suggested that the microbes could also cause disease, laying the foundation for medical microbiology. And after this, in 1865, George Mendel, he invented the law of inheritance. Actually, he gives the foundation of genetics. He conducted breeding experiments on pea plants in a monastery garden. His key discoveries were law of segregation. In law of segregation, he explained that each trait is controlled by a pair of alleles that separate during gamete formation, and he also gave the law of independent assortment. Genes for different traits are inherited independently if not linked. And he also gave a law of dominance. In law of dominance, uh, he explained that some alleles are dominant while others are recessive. Actually, his work published in 1865 laid the foundation of modern genetics. And after that era, Robert Koch, he gave the germ theory of diseases. He gave the foundational concept of microbiology and modern medicine. It established that specific diseases are caused by specific organisms, such as bacteria. In 1876, Robert Koch provided the first conclusive evidence that a microorganism like Bacillus anthracis was was the cause of a disease like anthrax. His work proved that microorganisms were not just present in sick organisms but were responsible for causing diseases. After this era, the Alexander Fleming, he discovered penicillin, the first true antibiotic. In 1928, while working with Staphylococcus bacteria, Fleming noticed that a mold, Penicillium notatum, accidentally contaminated a petri dish and killed nearby bacteria. The mold released a substance, penicillin, that could kill harmful bacteria without harming human cells. And his significance was that penicillin became the first true antibiotic, revolutionizing medicines and saving millions of lives. It laid the foundation for antibiotics era in treating bacteria. And large-scale fermentation for antibiotics and enzymes during World War II. In this era, developed to large-scale fermentation was developed to mass-produce antibiotics, especially penicillin, to treat wounded soldiers during World War II. The key advances in the era was shift from surface culture to deep tank fermentation. Aerated submerged culture enabled efficient large-scale production of antibiotics like penicillin, and its impact made penicillin widely available, saving countless lives in wartime. The third phase of modern or the third phase is considered as the modern or genetic engineering biotechnology. Its era is 1970s to 1990s. The major inventions in the era was the DNA double helix. The Watson and Crick gave the model that DNA is a double helix structure. Uh, Francis Crick, along with James Watson, they both covered the double helix structure of DNA, one of the most important scientific breakthrough of the 20th century. DNA is composed of two strands, twisted around each other like a twisted ladder. The sugar phosphate backbone from the side of the ladder, the nitrogenous bases adenine, thymine, guanine, and cytosine form the form the rungs. Base pairing rule, like the Chargaff's rule. The base pairing rule is regarded as the Chargaff's rule. Adenine pairs with thymine and guanine pairs with cytosine, held together by hydrogen bond. And these two strands run in opposite direction, 5 prime to 3 prime and 3 to 5 prime, and the replication mechanism, the structure suggested a semi-conservative model of replication where each new DNA molecule contained one old and one new strand. And after this breakthrough, recombinant DNA technology, invented in 1973 by Cohen and Boyer, gene cloning in bacteria. In 1973, Stanley Cohen and Herbert Boyer developed this recombinant DNA technology, marking the birth of genetic engineering. This technique allowed scientists to cut and paste DNA from different organisms and clone gene inside bacteria. In this experiment, the isolation of DNA, cutting DNA with restriction enzyme, and inserting that into the plasmid as a vector, and the transform and this the transformation was done into bacteria, and recombinant plasmid was introduced into E. coli bacteria. Bacteria expressed the inserted gene and replicated it, and creating clones. The significance of this recombinant technology was successful gene cloning in living organisms. It enabled mass production of proteins, for example, insulin, growth hormones, and vaccines, led to the development of genetically modified organisms, gene therapy, biopharmaceuticals, and the modern biotechnology industry. After, if, if we go next, the first genetically engineered organism was produced in 1974, and that was E. coli modified. In 1974, scientists created the first genetically engineered organism by introducing foreign DNA into the bacterium E. coli using recombinant DNA technology. The researchers like Stanley, who worked on Stanley Cohen, Herbert Boyer, and others, they inserted a recombinant
Sir,
plasmid.
sorry to disturb.
Yeah, yeah.
Some like your voice or again very
Uh,
very low. Yeah,
is it clear now? Is it clear now?
yeah, yeah. Now
Okay.
clear. Thank you so much.
Okay, okay. And if let's go forward, the first genetic engineered organisms, the researchers Stanley Cohen, Herbert Boyer, and others, they inserted a recombinant plasmid carrying foreign DNA. For example, the antibiotic resistant genes into E. coli. This created the first living organism with synthetic engineered DNA. Next, the if we talk about the next discovery, the monoclonal antibody. Monoclonal antibodies were invented in 1975 by Kohler and Milston. The first genetic engineering test was discovered in 1982, and human insulin produced by E. coli. The first transgenic animals in 1985, mice with foreign genes. A transgenic animal is one that carries a foreign transgene deliberately inserted into its genome, allowing it to express new traits or proteins not naturally present in the species. First transgenic mouse, the scientists inserted the foreign DNA, and the mice were used to study the gene function, disease models, for example, cancer, cystic fibrosis, drug testing, production of therapeutic proteins, and the significance. It opened the door for the genetic manipulation of animals, led to creating of disease models in mice, for example, Alzheimer's, diabetes diseases, paved the way for forming, producing human proteins, for example, clotting factors, antibodies in animal milk, and xenotransplantation research, gene therapy models, genetic engineer livestock for agriculture and medicine. Next, the PCR. PCR was invented in 1983. It transformed the concept of genetic engineering. Polymerase chain reaction. It was maybe the first who invented was the Carey Mullis. It revolutionized molecular biology, genetics, forensic science, and diagnostic, allowed disease gene detection, forensic DNA fingerprinting, genetic cloning and sequencing, and rapid detection of infectious agents. The next, this is the last phase, which is phase 4, and it's termed as post-genomic or omics era of biotechnology. This era started from 2000, and till now, the era of biotechnology is considered the post-genomic or omics era. The major invention till 2000 to until now, they are including like human genome project. The work on human genome project was completed in 2003. Full map of human DNA to sequence and map all the genes. It is almost 20,000 to 25,000 genes in human DNA, about 3 billion base pairs. The timeline, actually, this project was started in 1990, completed in 2003, ahead of schedule. Key achievements: produced the first complete blueprint of the human genome, identified most human genes and their locations, and it revolutionized genetics, medicine, and biotechnology, opened doors for personalized medicine, gene therapy, and disease gene identification. Next major discovery is CRISPR-Cas. It was invented in 2012. It is a gene editing technique. It's a revolution. Uh, in 2012, the scientists Jennifer Doudna and Emmanuel uh, Carpentier developed CRISPR-Cas as a tool for precise gene editing. CRISPR, a natural system found in bacteria to fight viruses. Cas9, an enzyme that acts like molecular scissor, cutting DNA at specific sites guided by a custom RNA sequence. Actually, this CRISPR-Cas allows scientists to cut, remove, or replace genes with high precision, easier transfer, and cheap than older gene editing methods. And this CRISPR-Cas impacted it revolutionized genetics with applications in medicines, agriculture, and disease research. Opened it opened possibilities for gene therapy, treatment of genetic disorders, and genome engineering. Next invention is it's the synthetic biology, The synthetic biology deals with the artificial gene circuits, synthetic life forms, for example, synthetic mycoplasma. In 2010, it was first done under the domain of synthetic biology. The field that combines biology, engineering, and computer science to design and build new biological systems or reprogram existing ones. This is synthetic biology actually. Actually, in this field, artificial gene circuits like custom designed DNA sequence that work like electronic circuits control when and how genes are turned on or off in a cell. And the organisms created or redesigned with synthetic DNA, all these things are under synthetic biology. The example is synthetic mycoplasma 2000 created by Craig Venter's team, first cell with completely synthetic genome capable of self-replication. And this synthetic biology impacted the applications in medicine, biofuels, bioremediation, and industrial biotechnology. It represents a new era in biotechnology where life can be programmed like software. And the next dis- the next discovery is CAR T cells therapy 2017. CAR T is the chimeric antigen receptor T cell therapy, a form of immunotherapy where a patient's T cells are genetically modified to better recognize and kill cancer cells. Next discovery is very important is the organized and 3D printing. In this discovery, uh, 3D bioprinting that is used in for disease modeling and regenerative medicine. Next, the current era in this current era, AI and machine learning is massively used in drug discovery, protein structure prediction. AlphaFold is a software that predicts the protein structure. AI analyzes massive datasets to identify drug targets, screen drug candidates, predict drug efficacy and toxicity. Actually, this AI speeds up and lowers the cost of developing new medicines. As a protein structure prediction, AlphaFold uses AI to accurately predict 3D protein structure from amino acid sequences, and it accelerates research in biomedicine, enzyme design, and genetic disease understanding. This is a short video. Please have a look on this. You will be able to understand the era of biotechnology. Sir, can you play this video? I can't play this video.
Uh, actually, this because it's YouTube live. So
Yeah, I can't play, sir.
yeah, it's YouTube live now. That's why video is not opening.
Ah,
So
okay.
we
Through.
will share we will
Yes.
share the slide into the group, so
Okay.
everyone can see there. Please
Okay,
continue.
sure. Yeah, sure. If I talk about the branches of biotechnology, the first branch, if I would like to discuss, is medical biotechnology. This is also termed as red biotechnology. Medical biotechnology is a branch of science that uses biological systems and organisms to develop solutions for human health, including disease prevention, diagnosis, and treatment. It encompasses the development of pharmaceuticals, diagnostic, gene therapy, and other innovative health solutions. The key development of medical biotechnology is the production of insulin using genetically engineered bacteria, mRNA vaccines for COVID-19, and gene therapy to treat genetic disorders. Next branch is agriculture biotechnology, which is also regarded as the green biotechnology, crop and livestock improvement under this domain. Biotechnology, agricultural biotechnology encompasses the use of scientific techniques, including genetic engineering, to improve crops and livestock. It involves modifying living organisms to enhance traits like pest resistance, nutritional content, and tolerance to environmental stress. This field aims to improve productivity, sustainability, and food quality in agriculture. And the third branch is the industrial biotechnology, which is also termed as white biotechnology. Industrial biotechnology actually uh, oftenly involves modifying or engineered microorganisms to perform specific tasks, such as producing desired chemicals of enzyme. It can also involve using naturally occurring microorganisms in fermentation processes to create various products. For example, microorganisms can be used to convert biomass into biofuels or to produce enzymes that enhance industrial processes. And engineered microorganisms can ferment sugars to produce a caprolactam, a key component of nylon. And like by ethanol production from sugarcane and using microbes to produce biodegradable plastics. The fourth branch is environmental biotechnology, which is also termed as grey biotechnology, and is bioremediation. The environmental biotechnology in the application of biological system including microorganisms to address environmental challenges and promote sustainability. It focuses on using biological processes to clean up pollution, treat waste, produce renewable energy, and develop sustainable technologies. And the next branch is marine biotechnology, which is also termed as blue biotechnology, and utilizing microorganisms to degrade or remove polluted. Marine is the field of science that deals with the ocean exploration for development of new pharmaceutical drugs, chemical products, enzymes, and other products and processes. It also deals with the advancement of aquaculture and seafood safety, bioremediation, biofuels, among others. This is also under this marine biotechnology. The technologies including platforms such as vessels, submersibles, observing system and sensors, communication technologies, and diving technologies that transport us across ocean waters and into the depths and allow us to scientifically examine, record, and analyze the mysteries of the ocean. The next is the bioinformatics, computational biotech, which is termed as yellow or golden biotechnology that has that is closely related with bioinformatics, focusing on applying computational tools to analyze and interpret biological data, while computational biology uses computational and mathematical approaches to model and study biological systems. The biotechnology is a broader field that utilizes biological systems for practical applications, including drug development, genetic engineering, and bioprocessing. The key activities under bioinformatics include the tasks like genome sequencing, protein structure prediction, data mining, and development of algorithms for analyzing biological data. Uh, the next branch of biotechnology is biodefense. This is a new concept. Like the biotechnology is also involved in biodefense, biowarfare. The biotechnology subfield. This is also called the biotechnology subfield of biodefense, encompasses the science and technology used to counter biological threats, whether naturally whether it's naturally occurring, accidental, or deliberate, and including surveillance, threat monitoring, medical countermeasures, and response planning. It also involves activities to prevent the misuse of biotechnology for offensive purpose, often terms often termed biowarfare or biological weapons development. The key aspects of biodefense or biowarfare in biotechnology is biosurveillance and threat monitoring, developing systems to detect and identify biological agents, medical countermeasure, research and development of vaccines, therapeutics, and diagnostic tools to protect against biological threat, biosafety and biosecurity. It actually establish protocols and practice to prevent the accidental or intentional release of dangerous pathogens and toxins. And it also deals with the counter proliferation efforts to prevent the development, acquisition, and use of biological weapons, and response and recovery, like the planning and preparedness for the responding to biological incidents, including public health interventions and emergency management. The examples are vaccine development, diagnostic development, antidotes, and antitoxins. Researching and producing treatments for toxins and infections caused by potential biological weapons. Next, I'm going to discuss one of the uh, technique used under biotechnology is PCR. Actually, PCR, uh, the term is the PCR means the polymerase chain reaction, also called as molecular phototyping, means a technique used to amplify small and targeted sequence of DNA to produce millions of copies of a specific gene fragment. This technique was developed in 1983 by Gary Mullis. He was awarded Nobel Prize in 1993 for his work in PCR along with Michael Smith. The application of biotechnology, you can see the PCR can be applied into many fields in diagnosis therapy, in nucleic acid detection assays, and there are several fields, almost all fields of sciences are has the use of PCR, which are dealing with microorganisms in respect. First, the conventional PCR, the general aim is to amplify DNA fragments using polymerase chain reaction PCR technique. The principal work is a PCR setup requires several components and reagents. The components include two pairs, two primers two pairs of primer, forward and reverse, and tag polymerase, TNTBs, buffer solution, distilled water, and template DNA. The PCR reaction follows the following steps: denaturation, primer annealing, and extraction. In denaturation, high temperature, almost 95 degrees centigrade, allows the separation of the DNA, the two strands of the DNA, due to the breaking or breakdown of the hydrogen bond, and the primers anneals. And the next step is primer annealing. The primer are short synthetic sequences of single-stranded DNA, typically consisting of 20 to 30 base pairs. Annealing usually takes place between 40 degrees centigrade and 65 degrees centigrade, depending on the length and base sequence of the primers. And the third part is the extension, that is also called elongation. The tag polymerase is a recombinant. In this, the tag polymerase is recombinant thermostable, that the DNA polymerase from the organism Thermus aquaticus. It is active at high temperature. The temperature is raised this stage to 72 degrees centigrade, and complementary DNTBs are added, starting the 3' end of the primer, and hence the tag DNA polymerase synthesizes in 5' to 3' direction. This is the overview of the PCR. Like, the PCR reagents are mentioned in this figure, and the thermocycler machine. The figure on the right side is showing the PCR reaction, like two strands of the DNA. In first step, the denaturation 94 degrees centigrade or 95 or 96, varies, and it denatures the double strand. It separates the double strand of the DNA, and in annealing, the temperature range between 52 to 68 degrees centigrade. Mm, and in this phase, annealing phase, the cooling, the reaction to allow primers to bind to their complementary sequence on the single-stranded DNA, and in the next extension and elongation phase, it raising the temperature to allow the DNA polymerase to synthesize new DNA strength complementary to the template, starting from the primers. And the cycling here, the cycles has a big role. These three steps, initiation, annealing, and extension, are repeated multiple times, typically 20 to 40 cycles. Mostly 35 cycles are also used to amplify the target DNA. These are the different types of PCR techniques: semi-nested PCR, multiplex PCR, RT-PCR, touchdown PCR, and inverse PCR. These are almost 32 types of the PCR that are used in laboratory, for scientific use. uh, I'm going to end this lecture. And if you want to ask any question, you are warmly welcome to ask any question. Thank you so much to all of the audience who listen this lecture.
Okay, thank you so much, Dr. Shahzad Zafar. Uh,
Thank you so much.
we are honored to have you 2 days here.
Yeah, thank you so much.
Okay,
Furthermore, I will share
uh,
this slide into the group.
yeah, sure. Uh, First, now, if anyone have any question, actually, thank you so much, everyone, to listening, and thank you so much in today joining us in first class. And I hope we demand your activeness and your passion for learning. And obviously, there will be proper attendance system, and like on the daily basis, like there will be 2 or 3 classes on weekly basis. Okay, so
Okay.
we demand your dedication, your passion for learning. It's a first class, it's general class, so that you can uh, take concept background of biotechnology. As a biotechnologist, you should know about the background of biotechnology. Okay, so it's Dr. Shahzad Zafar was actually giving you a lecture background of uh, biotechnology, and giving a lecture on some molecular techniques which are very important in the domain of life sciences, not in only biotechnology. So first, now anyone have any question, you can write your question, we will give you. And it will be better to make discussion at the end of session, so that everyone can clear those concepts, those their points, and their doubts in their minds. If you have any question, you can ask in chat box, we will respond you. Thank you so much, everyone.
Thank you so much, sir.
Thank you so much, Dr. Saab. We honor to have
You're
you
welcome.
in today's our class. And yeah,
Okay. Thank you so much. Yeah. Jazakallah khair. Inshallah, see you next time.
inshallah, sir. On Sunday there is second class. Uh, that class will be taken by Ma'am Amara,
So,
and
yeah.
is also a ex scholar at Tianjin University, China.
Thank you.
So
Yeah, she also served as a lecturer of biotechnology at UBAS, University of Biological and Applied Sciences, Lahore. Actually, its previous name was Lahore Medical and Dental College. Now it has become an open, a full-fledged university, and its name changed to Lahore University of Biological and Applied Sciences, UBAS, Lahore. And she also served there for 5 years. And she's also pursuing her PhD from Tianjin University, and she is also
so,
in.
sir, voice is very very low.
Yeah,
So,
she's also giving lecture tomorrow, and I would like to suggest to all of you to must attend her lecture. Thank you. You will get a lot of knowledge from her. You will seek a lot of knowledge from her.
Okay, maybe some mic issues to Sir Shahzad Zafar.
Oh
It's
yes.
okay. Yeah,
It's okay.
yeah. Obviously, at the end, like this internship domain include 18 lectures. Okay, so your attendance is mandatory in 18 lecture, and my voice is audible. Anyone, please let
Yes,
me
yes,
know.
yes.
My
Your voice
voice
is
is
clear
clear.
and audible. Yes. Yes, your voice is audible, sir.
I just want from. Okay, all right. Thank you so much. Actually, if I talk about lectures, there will be 18 lectures about biotechnology and bioinformatics. All will be half of the lectures, like 18, 8 to 9 lecture will be related to biotechnology, and 8 to 9 lecture will be related to bioinformatics. Okay, you will cover both aspects, and in future you will learn some gate skills like primer designing and some tools about bioinformatics. Just stay tuned. And for my first point is, your passion and your dedication is mandatory in this internship to make that summer program very blissful. Okay.
Inshallah. Okay, brother. Uh, can I leave now?
Yes. If anyone have any question, you can ask now. It's last chance. Yeah. Slide we will share in the group.
Yeah, inshallah. Yeah. I will share the slides, and there's a video. Must watch that video.
Yeah. In slide there is a video, live video. You can. Yeah. Internship duration. Did you not read out the PPT? Like we already give session on orientation session about all regarding all guidance about internship.
Sir Ghulam Murtaza, I also shared the PPT to you. You can also share the PPT in the group.
Okay, sure sir, thank you so much.
Yeah, you're welcome. Yeah,
Okay, now
yeah.
everyone can yeah, your attendance is already done, but it's confirmation. If you don't done attendance, fill this form, so Miss Canvi can take your attendance from this Google form. Everyone take make attendance on Google form. Thank you so much everyone for your cooperation. Any query about report? So report is mandatory for uh, certification and recommendation letters. So focus on your report. uh, no, if you are new one, actually this class is specifically for our internships. Those internees are selected specifically by a proper procedure. Okay, so I did share that link in other groups so that they can listen the lecture.